Antiviral properties of zinc. Pause text and illustrations for detailed study. Refer to constituent factors observed in viral diseases parts 1 through 5 for data relevant to this presentation. Definition of terms for illustration interpretation. Antigen presenting cell, APC. Ikappa-B kinase, IKB kinase, inhibitor of Kappa-B kinase. Interferons, INF, are specific signaling proteins released by host cells in the presence of a virus. The infected host cell will release interferons, causing adjacent cells to rally their cellular defenses. Interferon regulatory factors, IFN, IRF3, proteins regulating interferon transcription. Interferon sensitive response element, ISRE. Functions in interferon alpha-induced regulation of programmed death 1 PD1 in macrophages. Kappa-B kinase, enzyme complex propagating the cellular response to inflammation. Kinase, enzyme catalyzing phosphate group transfer from high-energy phosphate donating molecules to substrates with specificity in the process of phosphorylation. Macrophage M polarization, a process involving macrophages adopting various functional programs in response to microenvironmental signals. M2 macrophages regulate the resolution phase of inflammation and repair of damaged tissues. Mitogen-activated protein kinase, MEK12, MAP2K, MAPKK. Enzymes phosphorylate mitogen-activated protein kinase observed in signal transduction pathways. Mitosis, ordinary tissue growth involving cell division into two daughter cells possessing the identical number and type of chromosomes as the parental nucleus. Nicotinamide, adenine, dinucleotide phosphate oxidase, NADPH oxidase. Membrane-bound enzyme complex of the plasma membrane and phagosome membranes utilized by neutrophils to engulf microorganisms. Nidoviruses are a group of enveloped single and positively stranded RNA viruses infectious to humans and animals, comprising the Coronaviridae, Rhinaviridae, and Deltaviridae viral families. Nitric oxide is a cell signaling molecule. Nitric oxide synthase, NOS. Enzyme family catalyzing nitric oxide production from L-arginine. Nuclear factor Kappa-B, NFKB, protein complex controlling DNA transcription, cytokine production, and cellular survival. Nuclear factor of activated T cells, NFAT, transcription factors of immune system cells essential to their functional response. Protein NFATs also function in the development of cardiac and skeletal muscle as well as nervous systems. Oxidation is the gain of oxygen in the process of oxygen transfer. Reduction is the loss of oxygen in the process of oxygen transfer. Oxidation-reduction redox reaction, chemical reaction in which electrons are removed in the process of oxidation from atoms of the given substance oxidized and transferred to the atoms of the substance being reduced between chemical species. The species undergoing oxidation that loses electrons is the reducing agent, and that undergoing reduction that gains electrons is the oxidizing agent. If one substance is oxidized, the other must be reduced in a given reaction. Redox reactions are the main energy sources for biological systems. Molecular oxidation, resulting from hydrogen removal or combination with oxygen, liberates large amounts of energy capable of sustaining biochemical processes. Oxidative stress, imbalance attributable to excess reactive oxygen species or oxidants over the cellular capability to initiate an effective antioxidant response. An imbalance exists between production and ROS accumulation in cells and tissues and the capability of the biological system to detoxify the reactive products. Phosphatidylinosyl 3 kinases, PKT, enzyme family functioning in cellular growth, differentiation, motility, proliferation, as well as intercellular trafficking involved in cancer development. Polymorphonuclear neutrophils, PMN, granular immunoreactive white blood cells possessing enzymes released during allergies, asthma, and infections comprising basophils, eosinophils, and neutrophils. Protein kinase B, AKT, functions in cellular processes such as apoptosis, cell migration and proliferation, glucose metabolism, and transcription. Protein kinase R, PKR, an interferon-induced double-stranded RNA-activated protein kinase enzyme referred to as eukaryotic translation initiation factor 2 alpha kinase 2, functioning to protect against viral infections. Reactive Oxygen Species, ROS, oxygen-containing chemical species that are chemically reactive generated during mitochondrial oxidative metabolism and the cellular response to bacterial invasions, cytokines, and xenobiotics. They are byproducts of mitochondrial electron transport. ROS are unstable molecules containing oxygen that react with other cellular molecules. An excess of ROS in cell can be causative of damage to DNA, RNA, and proteins capable of causing cellular death. ROS are free radicals, existing as byproducts of cellular oxidative metabolism functioning in cell signaling, survival and death, differentiation, as well as inflammation-related factor production. Ribonuclease. RNase L, ribonuclease L. Enzyme catalyzing RNA breakdown into oligonucleotides and smaller molecules. RNA-dependent RNA polymerase, RDRP. Signal transducers and activators of transcription STAT are proteins involving the interaction of cytokines with their cognate receptors, functioning in the activation of latent transcription factors. STAT proteins are regulators of cellular differentiation, growth, and survival. Janus kinases, JAKs, function as intracellular non-receptor tyrosine kinases that transduce cytokine-mediated signals in the JAK-STAT pathway. The JAK-STAT cytokine receptor signaling pathway involves a chain of cellular protein interactions functioning in cell division, cell death, immunity, and tumor formation. This pathway activates the STATs that can bind DNA, allowing for the gene transcription involved in cell activation, division, recruitment, and survival. SIRT1, NAD-dependent deacetylase sirtuin-1, is a human protein encoded by this gene, functioning as an enzyme deacetylating proteins involved in cellular regulation. SIRT1 functions in metabolic pathways such as glucose metabolism, insulin secretion, affecting various metabolic substrates as a multifunctional protein. TIR domain containing adaptor inducing interferon beta, TRIF, adaptor that responds to toll-like receptor activation, organizing downstream signaling cascades for a specific cellular response upon exposure to a specific pathogen. Toll-like receptors, TLR, functional proteins of the innate immune system expressed on sentinel macrophages and dendritic cells that recognize structurally conserved microbial molecules. Transforming growth factor, TGF, is a protein cytokine cellular growth factor. Toll-like receptors initiate the innate immune response, sensing conserved molecular patterns for early immune system recognition of a pathogen. TRIF-Related Adaptor Molecule, TRAM, functions in toll-like cell receptor signaling. Tumor necrosis factor, TNF, cell signaling protein cytokine involving the white blood cells produced as a result of antigenic stimulation productive of a systemic inflammatory immune response. Zinc is an essential trace element to the active site of enzymes present at the tissue and intracellular levels. ZIRT, IRT-like proteins mediate zinc transport from the extracellular environment into the lumen of the cytoplasmic space, functioning in cellular immune responses. Zona occludens, ZO1, structural scaffolding proteins comprising the assembled multi-protein complexes at the cytoplasmic surface of intercellular junctions, connecting the integral membrane proteins with the filamentous cytoskeleton. Viral mechanisms of COVID-19 and how they might be opposed by zinc data illustration. Please refer to definition of terms as necessary. Involvement of zinc in various signaling pathways illustration. Zinc is a trace element essential to the development and function of the immune and redox system. Zinc homeostasis is essential to pro-inflammatory as well as regulatory immune system pathways. Zinc is important to the preservation of natural tissue barriers that prevent the entry of pathogens into the body. Disturbances of the respiratory epithelia allow for entry of viruses and co-infecting pathogens, leading to viremia and bacteremia, as well as other symptoms. Zinc is the most abundant trace metal in the body after iron. The functions of zinc are catalytic, regulatory, and structural. Zinc is believed to inhibit SARS-CoV RNA polymerase, affecting the degree of viral replication. Zinc is a functional component of various viral enzymes, proteases, and polymerases. Zinc functions in antiviral immunity and is essential to the generation of innate as well as acquired humoral responses to viral infection. Cellular and systemic zinc levels directly influence the body's capability to inhibit specific viral replication and dissemination. Metabolic zinc deficiency increases susceptibility to viral as well as bacterial infection. Zinc deficiency is causative of a decrease in the amount of functional immune cells, immune cell dysfunction, as well as an increased risk of infection, autoimmune disorders, and cancer. Zinc deficiency results in the impairment of zinc homeostasis, also playing a role in metabolic diseases such as cardiovascular disease, diabetes, and obesity. Zinc plays a role in the formation of stable cell membranes capable of inhibiting viral entry into the host cells. Zinc is an inhibitor of viral replication, interfering with the transcription of the viral genome, protein translation, polyprotein processing, as well as viral attachment and uncoding. Zinc may be an inhibitor of viral replication via alteration of the proteolytic processing of replicase polyproteins, as well as RNA-dependent RNA polymerase in certain viruses. Zinc may lessen the RNA synthesis of nidoviruses like COVID-19. Zinc possesses anti-inflammatory and antioxidative properties. The degree of the imbalanced immune response or cytokine storm presenting with systemic involvement and organ complications in advanced COVID-19 infection may result in part due to zinc deficiency. Zinc is a component of COVID-19 therapy at various stages of infection. Zinc is capable of preventing host cell membrane fusion with the virus, lessen viral polymerase function, impair the processing and translation of protein, inhibit viral particle release, as well as destabilize the viral envelope in Coronaviridae and other viruses. Zinc affects the mucociliary clearance of viruses, increasing ciliary beat frequency, enhancing viral particle removal, reducing secondary infection risk in the presence of the congestive lung pathology seen in advanced COVID-19. Zinc enhances the signal cascade of the T cell receptor and interleukin-2 as a second messenger. Zinc also functions in B cell maturation and function. SARS-CoV-2 is capable of infecting and impairing the function of B and T cells. Sufficient CD14 plus monocyte and natural killer cell quantities and cytotoxicity occurs in the presence of adequate zinc concentration. New presentations coming soon.